Hello, good day everyone. I am Sir Jeno, and for this lecture, we will be discussing the circulatory system. So let's get into it. So circulatory system is an organ system, which means it's composed of interconnected organs that work together to carry out particular function. So in this particular system, these interconnected components include the circulating fluid, which is the blood, the conduits, which are the blood vessels, and the pumping organ, which is, of course, the heart. So this system is concerned with the circulatory function. So circulatory refers to the mobilization or transport of materials all throughout the body. So these materials include nutrients, gases such as oxygen and carbon dioxide, hormones, and blood cells or corpuscles. So you have the RBC, WBC, and platelets. So basically, what happens in the circulatory system is that the pumping organ provides the necessary force or pressure to drive the circulating fluid in the vessels, and in the process, transport whatever materials are carried by the circulating fluid. So through this transporting mechanism, many bodily functions are achieved. So such as supplying nutrients and oxygen to the tissues, collecting metabolic wastes, fighting infections by delivering immune cells to the site of infection, repairing wounds, and maintaining overall homeostasis. Circulatory system is also like a superhighway connecting one organ system to another. Okay, so now let's look at this mind map to see how we will go about with our discussion. So we have three main branches here. First, we will discuss the components of the circulatory system, so the blood, the vessels, and the heart. And second, we will discuss the types of circulation. So basically, we have two main types, the open and closed. And then lastly, we will focus on the mammalian heart. So in the animal kingdom, there are many versions of the pumping organ. Some actually don't have a devoted pumping organ. So to limit our discussion, we will focus on the mammalian heart. So now let's move into the first branch, which is the components of the circulatory system. Okay, so most of our discussion will focus on this branch, on the components. So here we have three major components: the blood, the vessels, and the pumping organ. So because we will discuss a lot of things about these components, I will further give you a breakdown of the subtopics. So for the blood, we will be discussing the two major components of the blood. First, your plasma. Second is the corpuscles or the blood cells. So we have the RBC, the WBC, and the platelets. Next, we will be discussing the vessels. So we have three subtopics. First, we will discuss the arteries and arterioles. Next, the veins and the venules, and then the capillaries. And then for the pumping organ, which is the heart, we will discuss two major types: the chambered heart and the peristaltic heart. So the blood is composed of two major components. You have the plasma and the blood cells, or the corpuscles. So the plasma is a yellowish liquid that comprises approximately 55% of the blood volume. So it is the medium in which nutrients, gases, hormones, and wastes are transported throughout the body. So this plasma also have its subcomponents as shown here in the image. So subcomponents or the components of the plasma include, well, the majority is water, approximately 92%. Next, we have proteins. So proteins such as albumins, globulins, fibrinogen, and regulatory proteins. Yeah, so for albumin, main function for osmotic balance, transport of lipids, transport of hormones. For globulins, mainly for immune functions, but it can also work or function to transport ions, hormones as well, and lipids. Next, we have fibrinogen, which plays an important role in clotting. And then, of course, regulatory proteins. And then next, we also have solutes, other solutes, approximately 1% of the plasma. So these solutes include ions. So you have blood electrolytes that function to maintain pH by resisting change in pH, so a pH buffer. And then you have organic solutes, so lipids, carbohydrates for ATP production. And then next you have nutrients and gases and organic wastes. So these organic wastes, they include metabolic byproducts with no function. And these byproducts are toxic to the body if accumulated. So these, just to tell you something about these wastes, they are actually delivered to the liver and the kidney to be removed from the blood. So some examples of these waste products, actually they are nitrogenous in nature. You have urea, uric acid, creatinine, bilirubin, and ammonia. Okay, so next, the second component of the blood cells or the corpuscles. So this includes your RBCs, WBCs, and platelets. So these blood cells come about through the process called hematopoiesis or hemopoiesis. Hematopoiesis comes from the Greek words haima, meaning blood, and poiesis, meaning making or to make. So taken together, hematopoiesis means blood making. And tissues that are producing blood cells are referred to as hemopoietic or hematopoietic tissues. So in our younger years, approximately the first 25 years of our lives, hematopoiesis happens primarily in the bone marrow of tibia and femur. Afterwards, the hematopoietic process shifts to the axial skeletons, so that would include your vertebra, sternum, and the ribs, and become restricted in those regions. So all of blood cells arise from a single type of stem cell, which is here, we can see here as the multipotent stem cell, which is present in the bone marrow. So this multipotent stem cell has the capacity to differentiate into different types of cells. So this multipotent cell will first differentiate into two major lineages of progenitor cells. First, you have the lymphoid stem cells, and the other is the myeloid stem cells. So your lymphoid stem cells are produced in the lymphoid organs, and they give rise to the lymphocytes. So examples, you have the T lymphocyte, you also have the B lymphocyte, and you also have the NK or natural killer cells, which mainly function for immunity. The other major lineage, the myeloid, those that arise from the myeloid stem cell, you have here these blood cells. So first, myeloid stem cells are formed in the bone marrow. So example of the cells, blood cells that arise from this progenitor stem cell, you have your erythrocytes or your red blood cells, your platelets, your WBCs, so you have your monocytes, neutrophils, eosinophils, and basophils. So now let's zoom in to our corpuscles. So first, let's discuss the RBC or red blood cells. So red blood cells are also called erythrocytes. So erythrocytes comes from the Latin word erythros, meaning red, and cytos, meaning cell. So literally, it means red cell. And your RBC is color red because it contains the red protein hemoglobin. Hemoglobin is a protein which binds to molecules of oxygen and carbon dioxide. Your RBC has three unique cellular features. One, it's anucleate. That means it doesn't have a nucleus, and that is to accommodate more hemoglobin. Next feature is that it's biconcave, so it means it's a flattened disc, and its shape, as we can see here in the figure, and this biconcave shape increases the surface area for oxygen absorption. And last unique feature is that your RBCs are small and flexible so that they can fit through narrow blood vessels. Your RBC has a relatively short lifespan, approximately 120 days. So after which they age and die. And your RBC is actually the majority of your corpuscles, approximately 90% of cells in the blood. Okay, so here, the diagram here on the far right, it shows you the process of erythropoiesis. Okay, so recall, poiesis means to make or making. Now we append it with the word erythro to refer to the RBC. So erythropoiesis simply means the production of RBC. Okay, so this picture, this diagram shows you erythropoiesis, or the process of RBC production. Okay, so here, as we can see here in the diagram, your RBCs are produced in the bone marrow. And in this production, key nutrients from the food that we eat actually aid in erythropoiesis. So nutrients such as vitamin B12, folic acid, and iron. So you have the RBCs, they will be produced, they will mature, and then they will be released into the bloodstream to serve their function, that is to transport or deliver molecules. So after approximately 120 days, which is their lifespan, the old RBCs are removed from the circulation and transferred to the liver, where macrophages, your phagocytic cells, will break them down. So RBCs, some of its components will be recycled or used again by the body. So particularly hemoglobin. The hemoglobin of your RBCs can be broken down into its components. So particularly the heme and the globin. So the globin will be further broken down to its component amino acids, and these amino acids will be reused in the synthesis of new proteins. As for the heme component will be further broken down into iron and bilirubin. So the iron here will be brought back to the bone marrow to nourish hemopoietic tissues in the bone marrow. As for the bilirubin, it will be recirculated into the liver, bound to albumin, then stored in the gallbladder as bile. So bile will then be secreted in the small intestine to aid in the digestion of fats. So upon reaching the large intestine, bacteria will actually convert the bile into urobilinogen. And urobilinogen, when it's excreted, or when it's excreted through the large intestine, the urobilinogen will eventually be converted to stercobilin. And this stercobilin actually imparts the yellowish to brownish color to the feces. Okay, so some of the urobilinogen will actually be reabsorbed and will be excreted in the kidneys instead. And through that route, the urobilinogen will eventually be converted to urobilin. And this urobilin imparts the yellowish color of the urine. Of course, RBC production should also be regulated, and it is through homeostatic mechanisms that this regulation is achieved. So after 120 days in the circulation, old erythrocytes or old RBCs will rupture and die. It must be replaced by new erythrocytes through, of course, synthesis in the hemopoietic tissues. So if there are few RBCs in the system, few RBCs, we will expect that there will be few hemoglobin and thus lower oxygen levels in the blood. So this condition is called hypoxia. And hypoxic blood is detected by the kidneys and would prompt the release of what we call the erythropoietin, which is a substance that induces mitosis of progenitor cells of erythrocytes in the bone marrow. So with that substance produced, it will actually increase the RBC count, and as an effect, will also increase the oxygen carrying ability of blood, leading to the normal oxygen levels. Next corpuscle is the WBC, or the white blood cells. So white blood cells are also called leukocytes, from the Greek term leuko, white, cytos, cell. So taken together, literally, leukocytes are white cells. So as we've seen in the past slides, blood cells come from two progenitor cells, the lymphoid and the myeloid stem cells. Interestingly, for WBCs, you have cells that arise from lymphoid and some WBCs that arise from the myeloid. So WBCs coming from the lymphoid progenitor cell are the T cells, B cells, and the NK or natural killer cells. As for those WBCs that arise from the myeloid progenitor cell, you have the monocytes, which can develop into macrophages. You also have the eosinophils, basophils, and neutrophils. Now, if we will try to group them based on the presence of granules in the cells, you can group them into agranular or granular. So for agranular leukocytes, you have the lymphocytes, those that came from the lymphoid progenitor cell, and the monocyte. For the granular leukocytes, you have the basophils, neutrophils, and eosinophils. So in general, your leukocytes function for immunity. Specifically, of course, each type of leukocyte would have their specialty. So for T cells, their specialty is phagocytosis. So they would engulf invading cells. So maybe pathogens or foreign materials. For the B cells, they secrete antibodies. For NK cells or natural killer cells, they perform cytotoxic functions. So they can also control tumors and microbial infections. So for the monocytes, they are the largest WBC and can, again, as I mentioned, can differentiate into macrophages, which are also engulfing cells. They can also differentiate into dendritic cells, which can relay information to other leukocytes. So generally, your monocytes help in fighting bacteria, viruses, and other invading organisms. They can also help regulate adaptive immunity. Next, you have the basophils. Your basophils, they release substances that inhibit blood clotting, as well as chemicals like histamine that participate in allergic reactions. For neutrophils, this will be the leukocytes that prevent infection by blocking, disabling, digesting, or warding off invading particles and microorganisms. For eosinophils, they specialize on controlling parasitic infection. So your eosinophils, they converge on parasitic invaders and they release substances that kill that parasite. Next corpuscle, we have the platelets, or little plates. Your platelets are also called thrombocytes, Greek thrombos, meaning clot, and cytos, meaning cell. So literally, rough translation, clot cell. And that alludes to their function. It's because these platelets, these cells, are responsible for blood clotting or coagulation on cuts of the blood vessels. So platelets, they contain self-sealing materials that are activated when blood vessels are injured. So here, in the diagram, we can see the steps of coagulation. So when a blood vessel is cut open, it sends a prompt. So when that blood vessel is cut open, the body is exposed to potential invading organisms. And as such, the body is at risk of debilitating blood loss. As an adaptation, the body would respond by creating seals on the wound through the process of coagulation or blood clotting. So when the connective tissue is exposed, the platelets will first attach to the exposed endothelium, and then platelets will release the protein fibrin to impede the further leaking out of blood. The fibrin network traps the RBC and will completely seal the endothelium, as we can see here in the picture. We've established that fibrin, the protein produced by platelets, plays a vital role in the clotting process. However, a cascade of events must first take place before it can perform its function. In an open wound, platelets rapidly adhere to the damaged vessel and release platelet thromboplastin and tissue thromboplastin. This release of clotting factors with the aid of calcium ions will initiate the conversion of inactive prothrombin to its active form, which is the thrombin. Thrombin will then catalyze the conversion of the inactive fibrinogen to fibrin. So again, as we can see here, the release of fibrin or the activation of fibrin takes several steps. Now let's move to the next major component, which is the blood vessels. So first, we will discuss the arteries and arterioles. So here in the picture, you can see the anatomy of your arteries and arterioles. When we look at the structures, we can see that it is invested in elastic tissue, elastic fiber near the basement membrane. And along with this elastic layer, we also have smooth muscles and tough collagen connective tissues comprising the whole vessel. So this structure allows to withstand high pounding pressure of oxygenated blood leaving the heart and can accommodate surge of blood during heart contraction or systole and would return to its original form during heart relaxation or during the diastole. Main function of your arteries and arterioles is that they carry blood away from the heart. So you can use that as a mnemonic. arteries away from the heart. So arterioles are the smaller branches of arteries, but basically they perform the same function. They carry blood away from the heart. But for the arterioles, they also contain mostly smooth muscle for diameter adjustment. So these arterioles, they're expected to be farther away from the heart, so they receive relatively lesser pressure compared to your arteries. Next blood vessel, we have the veins and venules. So for veins and venules, they are made up of elastic fibers and smooth muscles, however, of thinner layers compared to the arteries. For their main function, they carry blood back to the heart. And so as a mnemonic, back, so V sounds like, and B, so they carry blood back to the heart. So a notable feature of veins and venules is that they contain one-way valves. So as we can see here in the picture. So this one-way valve is actually a flap of tissue that prevents backflow of blood. Since they carry blood back to the heart, you can expect that they receive even lesser pressure relative to the arteries. And because of that lesser pressure, there's a chance that they will backflow. And to prevent that, we have one-way valves. Most notably, your vein valves are found on the lower extremities. And that is expected because in those areas, there will be even lesser pressure. In addition, they're also surrounded by skeletal muscle pumps, only observed in the lower extremities. So when skeletal muscles contract, the veins are squeezed and the blood within them moves toward the heart. If not for these mechanisms, blood will accumulate in the lower extremities of a standing animal. Are you familiar with varicose veins? That has actually something to do with vein valves and lesser pressure. If skeletal muscles don't aid in the pumping of blood back to heart, the pressure causes deformations on the vein, causing skin bulges called the varicose veins. So when do you see varicose veins? During long periods of standing. So in those situations, the skeletal pumps are not activated. They can impinge on the veins to facilitate blood flow towards the heart. So to prevent formation of varicose veins, you need to do a physical activity. So you need to walk or jump or any physical activity to activate the skeletal pump to aid in circulation. Next type of blood vessel, we have the capillaries. So capillaries, they are minute links between arterial and venous systems, and they branch either directly from arterioles or venules, or they may branch from a thorofare channel called metarteriole. So this metarteriole, it runs in between arterioles and venules. So this metarteriole, also called the thorofare channel, contains precapillary sphincters. So as we can see here in the picture, these precapillary sphincters, they relax or contract, effectively controlling blood flow. So when the sphincters are relaxed, the blood from the artery will flow in all branches of the capillaries, as we can see here in the first picture. And this happens after having a full meal. This facilitates more efficient absorption of nutrients. On the other hand, when the precapillary sphincters are contracted, the blood will pass the metarteriole, this thorofare channel, and not on the capillaries, not on the branches. And this happens when we are performing strenuous activities. And that is because during strenuous activities, we have greater need for faster transport of oxygen and not absorption. That's why the blood is shunted through this thoroughfare channel so that it can be transported to the venules at a faster rate. In the capillaries or capillaries, or in these capillary beds, this is where the exchange of materials actually happens. Exchange of materials between the blood and the tissues. So this exchange is called capillary exchange or capillary exchange, and also called the bulk flow. So just to formalize the definition, capillary exchange, that's the exchange of materials from the blood into the tissues in the capillary. Majority of the transport or of the transfer of materials from the blood to the tissues and vice versa is through diffusion. In this exchange, there are two types of pressures at play. We have hydrostatic pressure and osmotic pressure. So hydrostatic pressure, that refers to the blood fluid pressure, which causes the outward movement of water molecules. That is seen here, or that is represented here as the yellow arrows. So this is the hydrostatic pressure. Hydrostatic pressure, hydrostatic pressure. Next is the osmotic pressure. So when we say osmotic pressure, that's the inward movement of water molecules due to high concentrations of plasma proteins in the blood. And that is represented here as the orange arrows. So this is osmotic pressure, this is osmotic pressure. In the arterial end of the capillary, so that is the portion of the capillary that's connected to the arteries, ultrafiltration occurs. So in this end, since blood pressure, since hydrostatic pressure is higher than osmotic pressure, you can expect that the net movement of water is out of the capillary. So with this net movement, it's actually leaving the filtered blood within the vessel. On the contrary, in the venous end, the opposite trend occurs. So in the venous end, blood pressure is relatively lower compared to the osmotic pressure. And because of that, the net movement would be towards the blood vessel. So in the capillary end, what happens here is reabsorption of water. So to summarize, arterial end, ultrafiltration, venous end, water reabsorption. However, this exchange only explains the movement of water and not of solutes, since capillaries are composed of layers of tissues that prevent movement of solutes. So for the movement of solutes, capillaries actually have openings. And these openings are, well, specialized for the movement of solutes. So they come in different forms. You have intercellular clefts, you have fenestrations, so you have small fenestrations and large fenestrations. It is through these openings that solutes from the blood move into the tissues and vice versa. Okay, so we've finished the first two components, the circulating fluid, which is the blood, and the blood vessels. Now let's go to the last component. We have the pumping organ. So we have the heart. So pumping organ is usually a muscular organ and is composed of contractile tissue. So when we say contractile, they are capable of contracting. And these contractions produce pressure that drives the circulatory fluid into the vessels. So in this discussion, we will discuss two major types, the peristaltic heart and the chambered heart. So first, let's discuss the peristaltic heart. For the peristaltic heart, it occurs in specialized sections of the vessels, wherein muscles in the walls of vessels contract in a moving wave that pushes fluid in front of it. By repeated cycles of this action, fluid is moved in one direction. This is the pumping method in many annelids, such as earthworms and many arthropods. Next, we have the chambered heart. Well, as the term implies, it's chambered, so that means it's compartmentalized. It means it have different sections with septa, with separations, with tissues separating one chamber from the other. So these chambers are generally divided into two. You have your atria, which are the upper chambers, and the ventricles, which are the lower chambers. So in the animal kingdom, there are modifications. So you have four-chambered heart, you have three-chambered heart, etc. For chambered heart, it actually requires one-way valves to prevent backflow of blood. Unlike the peristaltic heart, which can facilitate movement of blood in one direction, for chambered heart, there is a chance of backflow. And to prevent that, as an adaptation, chambered hearts also have one-way valves. Okay, so we've finished the first branch. Now we'll focus on the second branch, which is on the types of circulation. So for the types of circulation, we have two, the open and the closed circulation. For open circulation, it has a heart, a pumping organ, and vessels, but the blood mixes with the interstitial fluid. This mixture of blood and interstitial fluid is called the hemolymph, and it occurs in a space called the hemocoel. So for this type of circulation, it is commonly seen in arthropods in most mollusks. Next, we have the closed circulation. So for the closed circulation, it's more compartmentalized. The blood is separated from the interstitial fluid. So for this type of circulation, we can further divide it into two types, the single and the double circuit, closed circulation. This type of circulation is seen in vertebrates, annelids, and cephalopod mollusks. Now for the single circuit versus double circuit closed circulation, for the single circuit, so as we can see here in the first image, this is the type of closed circuit circulation seen in fishes. And what happens here is that literally the blood or the circulating fluid will make a single rotation, single circulation throughout the vascular system, where the blood is being pumped from the heart to the gills, to the respiratory apparatus, and where this oxygenated blood will then flow to be distributed to the body and then back to the heart. In contrast, the other circuit, the double circuit, which is seen mostly in mammals, the blood is pumped by the heart twice. So this system has two circuits, pulmonary circuit and systemic circuit. So what happens here is that you have your deoxygenated blood that will first pumped into the pulmonary circuit. So that is, you have here the deoxygenated blood, it will be pumped into the lungs, into the lung capillaries where it will be oxygenated, and then it will travel back to the heart. And then the heart will pump a second time, but this time it'll drive the oxygenated blood towards the systemic circulation, so towards the other parts of the body, delivering oxygen and nutrients to the systemic circuit. So here, of course, the body will receive the oxygen, will use the nutrients, and the blood that will leave those tissues will now be deoxygenated. And now they will return to the heart to be pumped again to the pulmonary circuit, and the cycle would continue. That's it for the circulation. Now we will focus on the mammalian heart. Let's first examine the anatomy of the mammalian heart. So of course, we're familiar with the chambers of the mammalian heart. Mammalian heart has 4 chambers. You have your left atrium, left ventricle, right atrium, right ventricle. But now let's look at the heart wall. You can see here in the picture, we have a transverse section of the heart. And if we zoom in on this portion, we can see that the heart wall actually has different layers. So in broad strokes, we have 3 layers: the pericardium, the outermost pericardium, the myocardium, and then the endocardium. So first, let's examine the outermost layer, which is the pericardium. Pericardium is also known as the heart covering. So the pericardium is a membrane enclosing the heart, consisting of an outer fibrous layer and an inner double layer of serous membrane. So the fibrous pericardium is a fibrous, dense, irregular connective tissue forming the outermost layer of the pericardial sac. And this anchors and protects the heart. As for the serous pericardium, it has 2 layers, as we mentioned before. You have the parietal layer of serous pericardium. This lines the pericardial sac, internal to the fibrous pericardium. So the layer immediately after the fibrous pericardium. Next, we have the visceral layer of the serous pericardium. It's also called the epicardium. So this visceral layer lines externally the myocardium. So this is a simple squamous mesothelium with connective tissues. So for these 2 layers of the serous pericardium, they both secrete serous fluids, which lubricates the membrane surfaces for easy heart movement. In between these 2 serous pericardium layers, we have the pericardial cavity. And this cavity contains a pale yellow fluid called the pericardial fluid. Next, we have the myocardium. So this myocardium, this is the thickest portion of the heart, and it contains the cardiac muscles. Inner to this layer, we have the endocardium. And this endocardium is a thin endothelium with connective tissue. So if we will look at an actual mammalian heart, we can see here the different layers. You have the outermost layer, the pericardium, you have the thick myocardium, and then you have the endothelial endocardium. Next, we will discuss the circulation of blood in the mammalian heart. So first, you have your right atrium. So the right atrium receives blood from the anterior vena cava and the posterior vena cava. The anterior vena cava, also called superior vena cava, drains unoxygenated blood from the upper organs. The posterior vena cava, on the other hand, it drains unoxygenated blood from the lower extremities. Posterior vena cava, or also called, or sometimes called inferior vena cava. Okay, so you have the unoxygenated blood from these veins. They would enter the right atrium. So from the right atrium, blood will then move to the right ventricle, to this lower chamber. So to prevent the backflow of blood back to the right atrium, we have a right atrioventricular valve, or the tricuspid valve. So this valve is functionally similar to the valves that we have seen in the veins. Whenever you encounter valves, at least in the context of circulatory system, valves would have the function of preventing backflow. Okay, okay. So blood from the right atrium, then it will go to the right ventricle. Now, here in the right ventricle, blood will move to the pulmonary arteries. Again, blood will, from this chamber, from the right ventricle, blood will move to the pulmonary arteries. So arteries, so they will carry blood away from the heart. So the deoxygenated blood here will be carried to the lungs via pulmonary artery. Okay, so this is the gateway to, or at least this is the gateway to the pulmonary circuit. Okay, okay, pulmonary circuit. So again, you have a valve that will prevent the backflow of blood, and that valve is a semilunar valve. So particularly, this is the pulmonary semilunar valve. Okay, so that's okay, here. Okay, so the blood will go to the pulmonary circuit, so to the lungs, and then from the lungs, it will go back to the heart via the pulmonary veins, okay, via the pulmonary veins. Again, veins carry blood back to the heart, okay? So just a review, right atrium, right ventricle, and then to the lungs via the pulmonary artery, so lungs, and then it will be carried by the pulmonary veins towards the heart, towards the next chamber of the heart, which is the left atrium. Okay, so the left atrium receives the oxygenated blood from the lungs via pulmonary veins. From this chamber, blood will then move into the left ventricle. Okay, so to prevent backflow, again, we have here a valve, and this is your left atrioventricular valve, or the bicuspid valve. Okay, again, left atrioventricular valve or bicuspid valve. Okay, so now we're on the left ventricle. So from this chamber, from left ventricle, oxygenated blood will now be pumped from this chamber to other organs. So in other words, from this chamber to the systemic circuit, it will now enter the systemic circuit. So the main vessel would be the aorta, okay? Aorta. So this is a large artery. And to prevent the backflow, to prevent the blood from going back to the left ventricle, again, we have a valve here, which is also a semilunar valve, but specifically here, this is the aortic semilunar valve. Okay, so the mammalian heart is, of course, in a double circuit. So in the previous slide, we have seen how the blood flows in the mammalian heart. So you have the unoxygenated blood or deoxygenated blood. It enters the right atrium, right ventricle, then to the pulmonary circuit via the pulmonary artery. And then in the pulmonary circuit, of course, the lungs is involved. So the blood will be oxygenated in the lungs, and then it will go back to the heart by the pulmonary vein. So this oxygenated blood will enter the left atrium, then to the left ventricle, and then will be pumped into the systemic circulation via the aorta. Okay, so here we can see in the systemic circulation, you have different tissues that are being serviced. You have different organs. You have the upper body, the liver, renal artery, kidneys, lower body, etc. So in this slide, I just want to emphasize that there is a unique case. So generally, the pattern is that you have your oxygenated blood that will be pumped into specific areas of the body, and of course, via capillary exchange, the oxygen will be delivered to the tissue, to the organ, and then after that, the blood is effectively deoxygenated and will be brought back to the heart to be pumped again into the pulmonary circuit. But one special case, one exception, is that there is this, what we call the hepatic portal system. You have this hepatic portal system. So instead of the deoxygenated blood being pumped back or being delivered back to the heart, it first enters a capillary bed of stomach, intestine, and spleen and pancreas, which are not directly pumped back into the heart. They are instead directed first into a series of veins, in capillary beds of the liver for detoxification before they are returned to the heart. Now let's discuss the cardiac cycle. So when we say cardiac cycle, we're referring to the alternating contractions and relaxations of the heart chambers. So it happens in a rhythmic sequence. So your cardiac cycle can be divided into two major phases. You have the systole and the diastole, and each of which subdivides into several smaller phases. So your diastole refers to the ventricular relaxation, so the ventricles are relaxed. And then systole, it refers to the ventricular contraction. So that's when the ventricles contract. Okay. So for the cardiac cycle, just some overarching principles. First, blood flows from higher to lower pressure. When contraction occurs, that effectively increases the pressure within the chamber. On the contrary, relaxation would lower the pressure. So, for example, we have here a diastole. So you have a ventricular relaxation. You would expect that the ventricles will have lower pressure relative to the atrial chambers. And if that's the case, the blood will flow into the ventricles. Okay, so next overarching principle is that your atrioventricular valves, so the valves in between atrium and ventricle, the AV valves or atrioventricular valves will open when atrial pressure are higher than the ventricular pressure, and closes when the pressure gradient is reversed. Again, so if the pressure in the atrial chambers are higher than the ventricular pressure, the ventricular pressure, then your AV valves are open, so facilitating the flow of blood from the atrium to the ventricle. Now, when the case is reversed, when the pressure is higher in the ventricles relative to the atrial chambers, then your AV valves are closed. Similarly, for the semilunar valves, they are open when ventricular pressure are higher. So when do we have that higher ventricular pressure? During, of course, ventricular contraction. So when your ventricular pressure is higher than the atrial pressure, the AV valves are closed, but the semilunar valves are open. So if that's the case, you would expect that the blood will flow out of the ventricles and into the aorta and pulmonary arteries. Okay, so for the cardiac cycle to occur, of course, you need to cause the heart to contract, and that is through excitation of specialized and modified cardiac muscle fibers. So in the myocardium, we do have specialized and modified cardiac muscle fibers, which specialized in a way that they generate and propagate electrical signals to stimulate rhythmic contractions. So mainly we have, or we will mention for excitatory tissues and fibers, we have the sinoatrial node, the atrioventricular node, the bundle of His, and the Purkinje fibers. So here in the picture, you can see the locations of these excitatory tissues and fibers. Okay, so first we have the sinoatrial node. So this sinoatrial node is also known as the pacemaker. It initiates the electrical impulses to stimulate contraction of the heart, and it's found in the atrial wall. Okay, so next we have the atrioventricular node. So this is this is the SA node, this is the AV node, or the atrioventricular node. So the atrioventricular node, it serves as an electrical relay station between the upper and lower chambers of the heart. So that is to say, your AV node electrically connects the atria and the ventricles. Next, we also have the bundle of His, or the atrioventricular bundle. So this is the bundle of His. So this bundle of His is a continuation of the specialized tissue of the AV node. So this bundle of His serves to transmit the electrical impulse from the AV node to the Purkinje fibers of the ventricles. Okay, now Purkinje fibers, here are the Purkinje fibers. The Purkinje fibers form a network of specialized cells that rapidly transmit cardiac action potentials from the atrioventricular bundle to the myocardium of the ventricles. So it mainly functions for coordinated ventricular contraction. Okay, so as for the whole excitation process, we have here a summary of that process. So first you have signals from the SA node that will spread through the atrial chambers. And then these signals will first delayed at the AV node. And then after that delay, specialized muscle fibers would pass the signals to the heart apex. And then the signals will spread throughout the ventricles. And when the signals spread throughout the ventricles, it would prompt the ventricular contraction. Okay, so just a lesson correlate. We have what we call the artificial pacemakers. So these artificial pacemakers, these are medical devices that uses electrical impulses to regulate the beating of the heart. So electrodes are actually attached at the apex of the heart for the blood to be pushed upwards to create the impulse to regulate the beatings of the heart. Okay, so we're on the last part. So the excitation of the heart is actually under nervous control. Okay, so both your sympathetic and parasympathetic nervous system influence your rhythmic contractions, no? Your heart rate. So two nerves link the circulatory or cardiovascular center of the brain with the SA node of the heart. That's how the nervous system affects, influences, controls the heart rate, the rhythmic contractions. So via two major nerves. First, you have the accelerator nerve. So the accelerator nerve is from the sympathetic nervous system, and when stimulated, it releases a neurotransmitter at the SA node that increases the heart rate. Next, we have the vagus nerve. The vagus nerve is from the parasympathetic nervous system, and when stimulated, it releases a neurotransmitter at the SA node to decrease the heart rate. So both nerves, both accelerator and vagus nerves, innervate the SA node and control the heart, but as we've discussed, they have opposite effects. Your accelerator nerves, when stimulated, it increases your heart rate. The vagus nerve, when stimulated, it decreases the heart rate. So that ends our discussion for the circulatory system. I hope you learned a lot in this lesson, and great job for finishing this lecture. Thank you very much.